100 in one institution.
Thanks, Kels. Can you hear me okay at the back?
Just.
Just.
It's a bit low.
It's a bit low.
Hold
okay.
it to your mouth.
I will.
You have to be like a rapper.
Righto.
Yeah.
Just my style.
No.
Okay. So, thank you for the opportunity to speak to you this afternoon. Yeah, my name's Steve Johnston from the University of Queensland. I'm presenting this talk this afternoon, I guess more as a discussion starter, just to also perhaps review where we're at in terms of some of the breeding of koalas that we've been doing in zoos in Southeast Queensland. Because Southeast Queensland really is the world's centre in terms of breeding koalas in captivity. We are the best at it, there's no doubt about that. So a little bit of patting ourselves on the back today, and also then perhaps also go on and talk a little bit more about where we could go as a zoo community. What would be the next step? What takes us into the next realm to make it really exciting? Okay, so let me begin my talk today by stating very clearly that I believe that the answer to this particular question that I've proposed is an emphatic yes. In fact, in our opinion as a group, and all those people there are our group, we believe that ZAAQ has already played a major role in this process already in terms of koala conservation. But it has the potential to play an even greater role, and perhaps a more hands-on role than it is at the moment. I have previously given this talk to a different group of people, a group of people that we did some work, or we did a talk at Central Queensland University, and there what we spoke about and what I directed my comments to were pretty much government officials or bureaucrats. And my job there was to really convince them that the zoos do have a real role for, in koala conservation. And I guess, as many as you know, some of those people can be quite difficult to convince. And some of them, in terms of those that have been in charge of koala conservation, have made our lives difficult at times. However, I'm very pleased to say that what we spoke about that day was received reasonably warmly. And it encouraged me to think that maybe there is a path forward that we can take. Because what we presented to them, I guess, was quite novel and also quite unconventional. So, the koala is Queensland's faunal emblem. So it's very natural that we should have a special affinity and responsibility for the koala's welfare and conservation. It is also the case that collectively, we as Queensland Wildlife Parks and Zoos have more captive koalas than any other region in the world. Enough, in fact, that we can maintain what is known by geneticists as a topic called, or a concept known as evolutionary potential. That is the ability to respond to selective pressure. And that's very rare that we'd have so many animals to be able to do that. I don't know what the current score is at the moment in terms of numbers of koalas, El? Is it but over 400, surely?
Holy moly.
Okay. So, this gives us some unrivaled opportunity in terms of what we can do as a community, as a zoo community, and how we can engage in the koala's conservation, how we can work with government to make this happen. And so that's what I really want to talk about. But first of all, let me just have a bit of a review. Zoos, of course, have already had a primary role in koala conservation in terms of disseminating koala-related information through public education and in being leading advocates of koala-related matters, both in the media and also in the general community. A good example of this engagement has been Dreamworld's commission and support of the Koala Land website, developed by Mr. Mark Gerarda and supported by Mucci. This webpage and blog that's associated with it is going to accumulate a vibrant online community and uses social media to gather and consolidate a range of social, political and scientific issues related to koala conservation. Empathy. Zoos also provide wonderful koala experiences for the general public and also to the international tourist in terms of allowing a very close-up encounter with the koala. This association often translates itself into life-changing empathy for the koala as a very special animal and also for its conservation plight. I, as you can see there on the left-hand side, as a 5-year-old, I certainly can attest to this love affair, as it would appear that my own early childhood encounter with a lone pine koala almost 45 years ago has no doubt had a lasting and perhaps ever subconscious impression and attraction for me. So in terms of funding, ZAAQ affiliated zoos have been very generous in providing funding for koala conservation through their commitments to the AKF and other various forms of koala research, either directly or via leverage through governmental schemes, like those run by the Australian Research Council and more recently by Queensland Government. For example, Lime Pine Koala Sanctuary and Australia Zoo have been working with the TIMS laboratory group at QUT in the development of a koala chlamydia vaccine. And our group has been fortunate to have ARC linkage partnerships with Dreamworld and Currumbin Wildlife Sanctuary for the establishment of breeding technology in the koala. We've also received the odd small amount of money from San Diego Zoo to conduct projects on male pathology, chlamydiosis, male chlamydia, look at male pathology in koalas. Most recently, we have also been fortunate, maybe you've heard, to obtain a state government koala disease grant that will incorporate and collaborate between a whole range of zoos, including Australia Zoo Wildlife Hospital, Currumbin Wildlife Hospital, Dreamworld and Moggle Koala Hospital. And these projects would not have been possible without direct involvement from the zoo community. And they shall examine things like the importance and incidence of chlamydial disease in male koalas, the effect of heat stress and on koala physiology and behavioural ecology, and also the social interactions of koalas and how this information might be used in models of koala disease. So zoos are making a real impact in that way as well. Queensland zoos have also contributed generously in terms of animals, in terms of zookeeper and veterinary staff, and they have allowed the description and the accumulation of a vast amount of fundamental biology on this species, including such things as reproduction, nutrition, behaviour and health. When we did the figures back in 2004, we estimated the cost of maintaining a koala, and I'm sure this has changed by now, was around about $6,000 to $8,000 a year. There's just no way that universities can be involved in that type of program without the help of the zoo community. And certainly we don't have that sort of money. Zoo enclosures have also been important testing grounds for new technology and also for the development of prototype devices, such things as GPS collars, vocal recorders and also proximity loggers. So we would test them in the zoo and then we'd go out into the field. And finally, how could we not mention the tireless and wonderful work of the koala hospitals, both at Australia Zoo and also at Currumbin Wildlife Sanctuary. In association with the RSPCA and Moggle Koala Hospitals, these institutions have provided world-class veterinary care for the welfare of koalas in Queensland, especially those in southeast Queensland. Certainly no one could deny that this work is not in the front line of the conservation battle and making a real and significant contribution to koala conservation. All of this is really important and extremely important and vital with regards to the facilitating koala conservation message. But we put it to you that it is possible for the zoos to do more, to be even more proactive, to take a more proactive role. Because of the large size of the captive populations that I was referring to, ZAAQ affiliated zoos are in a unique position to have a direct and pivotal role in the fine-scale management and genetic management and reproductive management of wild koala populations. We are therefore going to propose to you that zoos could function collectively as part of a live koala genome resource bank in which genetic deposits can be made or withdrawn. So this topic is not new, but it's one that we keep working hard at. Our group has now been successful in producing 33 koalas by means of artificial insemination, and the technique has now become quite routine across the industry, or for those that have participated with us. And in fact, earlier this year we helped in the artificial insemination production of a joey at Dreamworld, using semen from a male called Smojo. This animal is genetically very important but very shy and would not breed naturally. We were able to overcome this problem with the artificial insemination technique. Although we have been also successful in production of koala joeys using chilled transported semen, we haven't yet really cracked the biggie, which is to use frozen thawed sperm. That is sperm that has been frozen to minus 196, stored for some time, thawed out and then used for artificial insemination. We still haven't cracked that. And this remains the holy grail for us in terms of genome bank studies. However, there are still things that we can do where we can use the principles of genome resource banks. And that's what I'm going to talk about. So our conceptual model of a koala genetic exchange system or bank involves the use using koala zoo populations as a genetic reservoir. This can be used to maintain and store genetically important alleles from highly fragmented populations. Genes in the form of spermatozoa can be easily inseminated into captive females. We have the technology. These captive females can either be left on display or kept off display in quarantine enclosures and act as gene banks for valuable genetic exchange. Depending on their particular circumstances, animals from threatened or fragmented populations can either be bred naturally or by artificial insemination. ZAAQ affiliated zoos are the best in the world at producing and breeding koalas, and many are currently using the advanced reproductive management techniques for their own captive populations already. With just a few adjustments, the same efficiency could easily be directed towards the incorporation of genetics from the wild population. Yes, this is indeed a very controversial statement, but I assure you that it's possible to do. So, this is one form of genetic exchange. I've called it the genetic conductivity. So in this scenario, which we call genetic conductivity, we would use the zoo as a breeding centre by bringing in females from, say, wild fragment B into captivity for short periods of time and joining them with males from wild fragment A. Even better, we could collect semen from the males in wild fragment A, leave the males where they are in the wild, simply bring in the semen for artificial insemination. The female can then be subsequently released back into habitat B once a joey is old enough. And in fact, studies by D.G. de Villiers and others appear to suggest that Queensland koalas are in fact able to cope with these types of short-term excursions in and out of captivity. Have I gone too far? Genetic capture. In this situation, we would use the koala genome bank as to capture genetics that would otherwise be lost. One could imagine a scenario whereby koalas caught in the middle of a building or road development would require translocation. As we all know well, these situations do in fact arise and are currently topical and are likely to increase obviously in the future. Would it not be prudent in this situation that before the population is shifted and left to survive under its own devices, that we could capture a certain proportion of the important alleles from a genetically definitive number of males? This could then be done by collecting semen from the males during translocation process, or bringing the males into captivity for a short period of time, allowing them to breed with the females from the living genome resource bank. And this is a very simple, easy to implement strategy that would ultimately safeguard those important koala genetics. Genetic recovery. A very exciting possibility also exists whereby genetics would be rescued from animals that otherwise would be lost to both the captive and wild koala populations. There are literally hundreds or thousands, sometimes thousands of koalas that come into koala hospitals every year that have to be euthanized because of trauma or disease. The technology currently exists whereby spermatozoa from some of these koalas could be recovered and inseminated into living genome resource banks. We are currently conducting studies as part of the government funding that we received to determine the extent of chlamydosis in the wild male koala population. And it appears that although the prostate and the bladder may be infected with chlamydia, we can recover clean sperm from other parts of the reproductive tract, including the epididymis. And this sperm can also be used for artificial insemination. We're also exploring ways of recovering and collecting semen from diseased males and cleaning it up with anti-chlamydials prior to artificial insemination. In addition, semen can be recovered from uninfected males that are destined for euthanasia because of trauma or for associated with car or dog attacks. For these koalas, semen can either be electroejaculated from the anaesthetised animal before euthanasia or recovered from the epididymis of a post-mortem koala. And from the studies that we've done on koalas in the past, on wombats in the past, we know that if the animal is collected, if the tissue is collected relatively fresh and put into a refrigerator, it will last up to 3 days and still be suitable for artificial insemination. Genetic propagation, a fancy word for breeding. Perhaps I think the most breathtaking and significant role that zoos could ultimately undertake as part of a future koala conservation strategy is the genetically sanctioned production of koala offspring for release back into the wild.
True.
The Queensland Government has been accumulating koala habitat for a potential rehabilitation for some time. So would it not be an amazing outcome if zoos could be involved in the production of koalas to stock or restock these reclaimed habitats?
Fantastic.
There would of course also need to be other control measures put in place to reduce other threats such as dogs and cats, but under this scenario, zoos would be regarded as producers of wildlife, not consumers of wildlife.
Here, here.
While these proposals are bold and innovative, we nevertheless still need to be very cautious with respect to their ultimate application. Previous man-made manipulations of koala populations have not always been successful, as we well know, such as Kangaroo Island and French Island off the coast of Victoria. However, to be fair, many of these movements were conducted in a haphazard or an uncontrollable manner, without any consideration of the impacts of and on the koala. Recent studies by my colleague Jenny Seddon and her colleagues have started to establish a much better understanding of the genetic relatedness and diversity of koala populations in southeast Queensland, to the point now where we soon should be able to make genetically informed choices about the relationships between population fragments. This kind of genetic power means that koalas can be selectively bred to maintain genetic distinctiveness or to improve diversity of the population. These techniques will empower us as zoos to effectively manage the contributions that we make to this koala genome bank. Recently we had a paper accepted for publication which compared the genetic diversity of the Dreamworld and the Crumman Wildlife Sanctuary captive populations to those of their neighbouring wild populations. Genetic analysis of the captive populations revealed that both institutions had maintained the heterozygosity of their respective populations at a higher level than what occurs in the wild. This result is a credit to the stud book managers and also a testament to the industry to be able to self-manage koala genetics. Other issues that we will need to be cautious about, however, include things such as making sure that we don't have situations where we get the accidental spread of chlamydia or other genetically linked diseases that are associated by having a live genome bank. We have to be cautious about this, or that we produce animals that are not adapted or suitable for release back into the wild. So, in July this year, ZAAQ received its first round of funding from the Dreamworld Wildlife Foundation to develop a pilot program to explore the use of the live genome bank concept. Our first step in this project was quite a cautious one, was to determine the genetic diversity of the local Gold Coast Coomera populations. Using ear tissue biopsies collected by the wildlife hospitals, by EHP and also by the Gold Coast City Council, we will soon start to develop a thorough understanding of the genetic relationships and the diversity of the Gold Coast koala populations. We're currently holding back a little at the moment because, in terms of our analysis, because they're literally on the horizon. There will be a new set of genetic markers available for us that will be published that will allow us to get down to very, very fine-scale genetic management, allow us to make very fine-scale genetic management decisions. Once we have this genetic landscape all mapped out, we can then start to develop a particular strategy. And we can make a case to the EHP and say, look, let's have a go at a pilot program. We'll select a particular population on the Gold Coast and we'll focus on that population and we'll try and put some of these concepts into place. So, let me finish. Let me state unambiguously that the purpose of developing this technology is not to create a backdoor for the entry of new genetics into captive koala populations. Let me state that very clearly. I've heard this accusation from some that would oppose the process or the concept, but this is not and never will be our intention. Can I finish this talk by indicating that we are now in a position to offer ZAAQ affiliated zoos a commercial koala AI service, whereby we can deliver koala genetics throughout the state. This program would also need to involve training of zoo staff in female issues detection procedures. But we envisage that this could probably be managed quite nicely by means of a workshop. Mucci is of the opinion that such a program would have should have the blessing of ZAAQ, and I totally agree. I hope this is something perhaps that you guys might even consider or please talk about in your annual general meeting this afternoon. The specific costs and the details of such a program are still being worked through, but in principle it would essentially be a collaboration with those original collaborators, including UQ, Dreamworld and Currumbin Wildlife Sanctuary. We would need to recover our costs, of course, to deliver the AI procedure, but we'd also then, any additional funds we could use to put into koala research within the wild. Such a proposal would allow the sperm donor zoo to keep their male on site, but at the same time allow him to also participate in breeding off-site. We also believe that this program will help to facilitate the genetic transfer of koalas to smaller zoos that are currently struggling to maintain sufficient genetic diversity in their koala populations. I'll leave it there. Thank you.
Probably got time for a quick question if anyone has a quick one, but we're running a little bit behind time. Any questions? Mark?
Bloody fantastic.
Like obviously a fantastic program, very aspirational, great stuff. I think actually that might be where Grey Nose Sharks that may head in that direction as well. I'm not a geneticist, but I'm going to ask you a question anyway because I'm interested. You mentioned you took the sperm from a shy male and used that to inseminate a female. Is there a risk there? I mean, is that a heritable characteristic for a start?
Yeah.
And is there a risk there that you pass that on to the offspring?
Absolutely.
And my
there is.
question is, what tool can you apply to make the right choices about that sort of thing? And what tool are you applying?
Hey, great question. And yes, certainly from evidence in bulls and other species that libido does appear to be heritable, but it's not completely, the story's not complete in that. So even with species where they're domestic species, there's no clear evidence. But my gut feeling is that it can be heritable, that it's likely to be heritable. So yeah, we have to be very careful about those choices. The application of this particular technology on this particular animal was based purely upon a genetic decision in terms of diversity. So that's what we were going on. But you're absolutely right on the money that if we're going to apply those sorts of principles, then there's going to be other variables that should be coming into the mix in terms of making choices about the animals. So susceptibility to disease could be one of those, etc., etc. So at the moment, what we're working through is the mechanics, if you like, of the process. Our decisions at the moment are based upon genetic ones only. But we'd be fools not to have an understanding of the other processes that are going to impact.
All right. Thanks.
No worries.
Thanks very much, Steve. Most of you would know Steve has worked with... Didn't want to hear me. Most of you would know Steve has worked with a lot of our different facilities over time and has always been fantastic in presenting his work to us at the conferences. So thank you very much again, Steve. Our next presentation is from Ellis Swinberg, and again collaborating with Steve and with Tina Jensen with regards to southern hairy-nosed wombat breeding. Again, Tina has been affiliated loosely with many, there she is over there, with many zoos through time and has done some great recovery work with bridled nail tails and working towards northern hairy noses as well. And Elise is going to talk to us about some conditioning she's done at Tina's facility, conditioning southern hairy-nosed wombats for urine sample collection. Thank you very much.
I was going through this at home and I thought maybe it'd go a bit too long, but after hearing Graham's presentation, I have tons of time.
We are running behind time now.
Good afternoon, my name is Elise Swinburne and I'm a PhD student with the University of Queensland, and today I'll be talking about conditioning southern hairy-nosed wombats for regular urine collection. Why not? So as you would be aware, the northern hairy-nosed wombat is critically endangered with less than 200 individuals in the wild. If we are to establish a successful captive breeding and breeding for release program, then we need to have a thorough understanding of this species' ecology, social structure, reproductive behaviour, biology and endocrinology. Unfortunately, there isn't a lot of information available and more research needs to be conducted in these areas. Now, if the northerns are critically endangered, why would we be working with southerns? Well, there are no northerns in captivity and the southerns are morphologically similar. We believe that they are a good model for northern reproductive behaviour and endocrinology. Sorry. In any event, the southerns are currently not endangered, but they are in decline, and it may be necessary that one day in the future we will need to breed this species in captivity, which traditionally has been very difficult. Success is not unheard of, it is just not guaranteed, and it is not as common as we had hoped. Therefore, a better knowledge of this species would be beneficial for any captive breeding program. Current collection methods for endocrinology and reproductive research rely on methods such as plasma collection, which can be highly invasive and stressful for the animal. These invasive and stressful techniques can have negative impacts on the reproductive success in captivity, which is obviously counterproductive. Previous wombat research has focused on fecal hormone analysis for information regarding reproduction. However, due to the 3-day lag time from digestion and the fact that it is difficult to measure estrogen and impossible to measure luteinizing hormone, fecal hormone analysis does not allow for accurate timing of estrus. Urine offers a non-invasive means of collection and can be analysed for both protein hormones such as luteinizing and follicle-stimulating hormone, as well as steroid hormones such as estrogen and progesterone, allowing for more up-to-date information regarding key estrus events such as follicle maturation and ovulation. The information regarding the urinary information can be used for diagnostic purposes, but it can also be used to enable accurate timing for procedures such as artificial insemination. Of course, this raises the problem of how do you collect urine from a wombat? Fresh samples are preferred as scavenged samples off the ground or den floor can be contaminated with bacteria, urine or fecal material from other animals such as rodents or den mates. Therefore, the problem that we are attempting to overcome is how do we non-invasively collect urine samples from a southern hairy-nosed wombat. So my research, the research that we are conducting is just one part of my PhD, which is investigating the reproductive behaviour and urinary hormone profiles of the southern hairy-nosed wombat. The intent is to correlate the hormone profiles with the reproductive behaviour in order to determine non-invasive methods of detecting estrus in wombats. We would like to obtain fresh urine samples. Therefore, we have developed a process of conditioning these animals to provide a sample on a regular basis. So the initial process was developed by Tina Johnston at the Australian Animals Care and Education Facility, and this was initially developed for toileting of hand-raised pouch young. So this is the Wombat Research Facility at ACE in Mount Larcombe, 30 kilometres west of Gladstone. It currently houses 18 southern hairy-nosed wombats, divided into 8 enclosures, each housing either a pair or group of mixed or same-sex animals, depending on their specific research criteria. Just out of interest, ACE is also heavily involved in the bridled Nautau wallaby captive breeding and breeding for release program. And we are also the main centre for rescue and rehabilitation of native wildlife in central Queensland. We currently have pythons, gliders, cockatoos, cygnets, and we've also got a couple of wallaby joeys. So there is never a dull moment at Mount Larcombe.
Wow.
So the animals used in this trial were a combination of hand-raised as well as adult wombats that came into Tina's care. There was a total of 7 animals that were chosen for the conditioning trial, and these first 5 were chosen for tactile conditioning based on their temperament when interacting with humans. They tend not to bite you. Please note the word tend. My boots have been bitten by 4 of these females. All of these females came into Tina's care as either pinkies or juveniles before the age of 2. And these top 3 had actually been previously conditioned for toileting as joeys. These 2 females came into Tina's care as adults and had pre-existing behavioural issues such that they were very timid and did not respond well to tactile conditioning. Therefore, a different approach was required. We used a false floor instead of tactile conditioning. So this is the board, and we can easily slide a tray underneath that board to collect a fresh sample each day. Okay. Now, unfortunately, it is not practical to follow a wombat around with a bucket hoping to catch a sample at the right moment. They don't. Firstly, I'm a PhD student. I have other things to do during the day. And secondly, wombats don't take to kindly to having strangers in their yard following them around. You will be bitten hard. So, how do you condition a wombat to give you a sample when you want them to? The first stage in this process is habituation and observation. This is the period when the animals are given time to get used to my presence in their dens. This was achieved through daily husbandry and feeding, as well as enabled me to observe their natural elimination behaviours. Prior to undertaking this trial, Tina Johnston and Peter Brooks conducted the majority of the husbandry. However, for me to undertake this trial, we gradually transitioned for me to take over a more significant role. Their presence was integral for the animals to get used to me. The second phase is conditioning. So from the observation phase, I observed the characteristics that wombats tend to urinate shortly upon waking. This represented an opportune time to collect a fresh urine sample. Therefore, upon waking, I would apply a tactile stimulation of scratching the animal just above the tail until it urinated. I conducted this twice a day at about 6 o'clock in the morning and then again in the afternoon. I timed the period from waking to urination, and upon urination I would give the animal a reward in the form of a firm rump scratch. Okay, the third stage is collection. Now, it is all well and good to condition a wombat to urinate. However, without collection, all you end up with is a puddle on the floor. Part of the habituation process was to condition the animal. Part of the conditioning process, was to habituate the animal to a small collection tray. In this case, a single egg frying pan. You'll never cook out of one of these again. This device was used for several reasons. Firstly, it is nice and low and can easily be slid under the animal's rump. Secondly, it has a non-stick coating, which means the samples can be collected clean and the pan itself can be easily cleaned between animals to prevent cross-contamination. And thirdly, it has a nice long handle, so I can hold onto the pan while the animal is urinating, keeping my hands out of the way. This device holds about 150 ml, suitable for even the largest wombat's bladder, and best of all, it cost me $3 from Woolies.
Wow.
Okay. Oh, your thing's broken. Brad. I'm going to have to sew again. Okay, we'll start that again.
Yes.
Okay. So this is This is a video of non-invasive collection of urine collection. This is Holly, and she's part of our induced ovulation trial. We're determining whether they cycle without the presence of a male. So she's in there with another female. If it's not going to work, I can just describe it.
You describe or try.
Okay. So this is Holly, and I observed that she would urinate prior to feed time. So before that, I would withhold her food until we conducted collection. So of an afternoon, I would go in and I would wake her up, and I would slide the collection tray under her rump, and I would apply tactile stimulation until she actually urinated. Upon urination, I would then give her a firm rump, some verbal praise, and then I would also give her her food. This video test takes about 45 seconds. That's how quick it took. So it was just demonstrating just how simple this process can be when it works. Cool. That's right, we can go to the next slide.
Much better than I do. Wonderful.
Is that the next one?
Yeah, that's the next one. Yep. Okay, now everyone here would know that animals are not robots. And there was variability between the time to urination, the amount they urinated, whether or not they urinated when I wanted them to, and whether I was successful in collecting a sample. This first box demonstrates this is the percentage of animals that responded to tactile stimulation. As expected, the numbers increased from around 50% at week 5 to nearly 90% by week 16.
Wow.
This second box shows the raw time to urination, which is better demonstrated in the following graph, which shows a gradual decrease in time from around 7 minutes at week 5 to around nearly 2.5 minutes by week 16. After week 5, there was only a gradual decrease. There was only a marginal decrease in time. So whilst the previous slide showed the overall time to urination, there was variability between the individual animals. For example, the majority of the animals responded quicker in the morning compared to the afternoon. However, for one wombat, there was a substantial difference between the AM and the PM collection, and obviously Wiggles is not a morning wombat. Indeed, all animals exhibit their own behavioural characteristics, and it is perhaps one of the most important aspects of habituation and conditioning for the researcher to thoroughly understand their animals. For example, now this is another video, I hope it works. No. Okay, it worked this morning, I swear it worked this morning. Talk amongst yourselves.
Okay.
I'll just talk now.
You've broken my machine.
Broken machine.
Is it now not working anymore?
Okay, we've done pretty well so far.
Now we're definitely going to run over time.
Sorry. That's alright, I'll blame Brad. We usually do this.
That suits me fine. Okay, good. Okay. For example, Amy would urinate in den 1. Den 1 is this one on the right-hand side. At all other times, except urine collection times, this den was closed off to her. So when the den door was open and she was able to access this den, she would immediately urinate. Now in this den system is her male, Foxy, and he's quite large, and there is absolutely no way I would enter this den system if he wasn't locked down. He would eat me. So upon urination, I give her a firm rump scratch, and then I'd let her outside. Urine collection is also a good time to collect fresh fecal samples with your bare hands, so come shake my hand up. I think what we did was we didn't close it down. Okay, so Hope and Bella here would also have very different collection routines. Hope in the centre here, she would urinate after I cleaned her den and her false floor. So this would ensure that I collected a fresh urine sample. Bella, on the other hand, would urinate urinate upon waking. So I would need to ensure that I was ready for immediate collection, otherwise she would sit in it or urinate on the den floor, potentially missing or contaminating a sample. And this one's another video. It's working. Some wombats also have multiple toilets, one inside and one outside, and at times I would have to venture into their yard enclosures to collect a sample. Holly here would wake up about an hour before dawn and an hour before dusk. So I too needed to be up at those times ready for sample collection. It's easier collecting an hour before dusk than it is at dawn. Collection is not a straightforward process of sliding the tray under the animal and waiting for them to urinate. The animals do move around a lot and they do make it difficult sometimes to collect from.
We've all been there.
for wombats tolerate human presence when urinating. One of the females, Isa, actually had to be removed from the trial because she became quite aggressive towards me, and urine collection became quite stressful for the animal, but more so for myself. Some animals also took longer to get into a regular collection routine. For example, Bella took 31 days before I could collect consecutive samples from her. Amy took 40